everyone, welcome back. So we're in our 7th week and we're talking about Mendelian genetics. So basically how we get from genotype to phenotype. And if you take a look at the image that we have here, we have a bunch of twins in this image. But one thing, if you know any twins, is that they are not exactly the same, right? They are not the same person. They're not a clone of the same person, even if they come from the same egg, right? So you have twins that can come in different variations. So you can have one egg that actually, after it was fertilized, divided and then became two individuals, right? And then you can have two eggs that were each fertilized and became two individuals. And so completely separate genome. But even when you have one egg that is separated, these are identical twins, and become two individuals, they are not exactly the same person. And that's because we have other ways that our phenotype can be expressed, even though the DNA may actually be the same. So let's take a look at how that can happen and the underpinnings of understanding genotype to phenotype. And this happens around the world, all the organisms that we see, that we can see this expression of the phenotype. And remember, phenotype is anything that you can see, anything that you can measure, that is the expression of the genotype. And we know that even though we have two individuals who may be coming together and creating a variety of offspring, each of those offspring are going to be very different. So let's explore how that happens. So the inheritance of genes. So here we have, we're looking at the maternal chromosome pair, that is the chromosome pair from the mother, and we have the paternal chromosome pair, that is the chromosome pair from the father. And here we're just going to be looking at one gene. So remember that humans have 23 pairs of chromosomes. That is 46 individual chromosomes. 23 is the haploid number, and 46 is the diploid number. And so thus we have two copies of each gene. When we basically have one of the copies of the gene end up in the egg and one copy of the gene end up in the sperm, that's how we have our haploid egg and sperm. When they come together, then the gametes unite during fertilization, and the child inherits one set of chromosomes from each parent, and thus two copies of each gene. So let's just kind of look at why we care about inheritance of genes. Well, there are some people that have this syndrome that you may have never heard of, but it greatly affects their life. This is fish odor syndrome. And it's also, its scientific name is trimethylaminuria. And it's a genetic disease. Symptoms are often present from birth. And fish odor syndrome is characterized by this offensive body odor. It's offensive to the human. Offensive body odor and the smell of rotting fish due to excessive excretion of trimethylaminuria, TMA. And it's excreted in the urine, the sweat, and the breath of affected individuals. Well, as you can imagine, this would be very difficult to go through life with that, right? But let's take a look at how that happens. So here we're looking at the maternal chromosome pair. Here we have the mother that has the normal version of the FMO3 gene, and a defective version of the FMO3 gene is responsible for fish odor syndrome. Okay? And so now we're looking at the paternal chromosome pair, and here we see he has one normal version of the gene, and he also has one abnormal version of the gene. Well, now when we look at the maternal gametes, the eggs, she can either make an egg that has a normal version, or she can make an egg that has an abnormal version. Same thing for the paternal gametes. He can either make sperm that have a normal version of the FMO3 gene, or he can make sperm that has an abnormal version. Well, what can happen is if we have an egg with an abnormal version and an egg with a sperm with an abnormal version, when those two unite, well, now this individual has two abnormal versions of this gene. And if the child inherits two copies of the defective version of the FMO3 gene, then the child will develop fish odor syndrome. Okay. So once breeders actually recognized the existence of heredity, selective breeding began. And such as for reduced size in horses became possible, and as you can see here by this miniature horse. And so we learned that we can control the phenotypes of other organisms. Here we're looking at some examples of phenotypic control that we have asserted on other organisms. So we're looking at cats. Here we're looking at fur length in cats. This is an example of a single gene trait. It's occurring on a single gene, so the gene for fur length. So you can have long hair or you can have short hair. The gene for coat color is also a single gene trait in cats, so we can have a gene for white hair on a cat, or we can have color on the hair of the cat. And so we have selectively bred for cats, dogs, horses, cows, turkeys, you name it. We have selectively bred for different organisms that we prefer. But it wasn't that long ago, it was actually through the 1800s, that humans didn't understand how heredity actually worked. And they had this mistaken idea that a tiny pre-made human existed in every sperm cell. And that theory was introduced in the 1600s. So that hypothesis actually was introduced in the 1600s. Is very, very wrong, but it remained popular through the 1800s. And that was what was believed as to how heredity happened. But then along came Mendel. And Gregory Mendel did research with peas. And if you want to read a very interesting backstory, because sometimes the backstories of science are fascinating, read the backstory of Gregory Mendel and how he actually came across breeding peas, and even how he even started using peas as the model organism. That is a very interesting story, and it kind of highlights how a lot of the discoveries, like the major sort of earth-shaking, paradigm-changing discoveries in science, kind of happen by happenstance. They end up being sort of happy accidents where it wasn't planned to do X, Y, and Z, or even to use peas, but because circumstances were such that that's what he had to use, they ended up being the perfect organism to study. We're going to see why that is. So let's talk about why peas ended up being the perfect model organism. So number one, they're easy to maintain. They're very easy to breed. They reproduce very quickly, creating multiple generations to observe. They have variants in traits such as flower color. There are different variants, purple and white flower color, variants in their pea shape. So they're round peas and wrinkled peas, and variants in the pea color. So you could have green peas and yellow peas. And so they have lots of these traits that can be used to observe and determine what is happening genetically. And so what Mendel did was he created these established populations, these very distinct populations. So he used true breeding plants. They always produce offspring with the same variant of the trait as the parent. So there's no variation down the line, and that's called a true breeding plant. So for example, if you had a true breeding purple flower plant, the parents had purple flowers, also the offspring of those plants also had purple flowers. Same thing if you had a true breeding white flowered plant, then the parent plant would have white flowers and also all the offspring would have white flowers. So you had no variation that would creep in. And that just made it really the perfect organism to study genetics. And so what you're going to learn about is what's called dominant and recessive traits. How these different traits actually come to be expressed. So here we have what Mendel did. So he crossed true breeding purple flowers of plants with true breeding white flowers of plants. So when we see this X, that means that he crossed them, he bred them. And in plants, that means you pollinate them, right? So he bred true breeding purple flower plants with true breeding white flower plants, and he noticed that the purple color is the dominant trait, while the white colored flower is a recessive trait. That means whenever that purple trait is present in the offspring, it's going to be the trait that is expressed phenotypically, right? Even though the white flower gene may be there, okay? That is the dominant and recessive traits actually being phenotypically expressed. So the recessive trait for the white colored flower must have been lurking somewhere in the previous generations, even though it was not visible. So when he bred this true breeding purple flower plant with the true breeding white flower plant, he saw that the second generation, they all had purple flowers. But when he crossbred the second generation to each other, then suddenly the white flowers popped out again, that he started to see offspring that had white flowers. And that's where he began to understand that the gene for the white flower was there, it just was not being expressed. Okay. So we have what's called Mendel's law of segregation. And according to Mendel's law of segregation, only one of the two alleles for a gene is put into a gamete at fertilization. And offspring receive one from each parent, okay, for a gene. So here we are looking at the illustration of that. So here we have what's called a heterozygous pea plant, which means heterozygous means that it has two different alleles, okay? So we can see, and they're using coloration here. The lighter color is signifying one version of that gene, and the darker color is signifying another version of that gene. So alleles, right? Two different alleles. We also have the term, so that's new terminology for you, heterozygous, which means two different versions of the same gene. But when we cross, when we're looking at two different parents that are both heterozygous, we, through the process of meiosis, which you learned before, each gamete gets one copy of the gene, right? Because we're going from diploid to haploid, right? And then at fertilization, then they each end up with both copies, one from each parent, right? That's exactly what we already know. We've gone over it 1,000 times, but it has a name. And so this is where you're learning the name. It's called Mendel's law of segregation, where we actually see that only one of the two alleles for a gene is put into the gametes. He's the first who came up with that. All right, so I've been using these two words quite often. They should be very familiar to you. But here, when we're using terminology, when we're using the terminology of genotype, we would say, looking at these two deer, if I'm speaking of the genotype, I would say that this deer here on the right is homozygous, which means it has the same two alleles. They are the same. Homozygous for the recessive allele for albinoism. Okay? So, translation, homozygous has the exact same alleles, two alleles, for the recessive, meaning that, that there is no dominant gene that's present there, because if it was a dominant gene, it would be expressing. And so this is a recessive gene, and it's expressing the recessive gene because it's two recessive genes. And that's why it is an albino deer. So when you're speaking of genotypes, when you're speaking of the genome, there is language for that, right? Homozygous for the recessive allele for albinoism. When we're speaking of phenotypes, this is what your more commonly used language. You're familiar with that. Speaking of the phenotype, so the phenotype of this deer would be little or no pigment in the eyes, hair, and skin. Okay, so I want you to get extremely familiar with these two terms and the terminology that is associated with each one. So now we're going to be talking about genotypes, where we're actually speaking of what's happening on the genes in the genome, and we're going to be talking about phenotypes, what we can see expressed physically. Okay. All right, so we have something called the Punnett square, and Punnett squares actually help us to understand the percentages, the probability of different offspring if we breed two different individuals. So the Punnett square can actually help us go from the genotype to the phenotype and understand what that is. So here is the Punnett square. It's a very useful tool for determining the possible outcome of a cross between two individuals. So here we have the mother giraffe, and she is phenotypically albino, and genotypically she is homozygous for the gene little m, okay, signified as little m. So if we're looking at her genotype here, we would say little m, little m. And then we have the father here as we set up our Punnett square, and he is pigmented, that is his phenotype, and his genotype is homozygous dominant M, okay, signified by capital M, capital M. Now, by convention, when we are writing out genotypes, we always write the recessive genotype in lowercase, and we always write the dominant genotype in uppercase, capitalized, okay? And that helps, and it's good that that is the way that it's done by convention, because when you're just looking at it, and this is going to become easier for you, you'll just be able to look at a genotype and immediately know, when I see two lowercase m's, I know immediately that this is homozygous recessive. And when I see two uppercase m's, I know this individual immediately is homozygous dominant, right? If I saw a individual that was written with a capital M and a lowercase m, I would immediately know that it is heterozygous for that gene, right, because of that convention. So that's very important. So in the Punnett square, when we're looking at a gene and we're trying to figure out, determine the outcome of a cross between these two individuals, here we have one individual that is here at written at the top, lowercase over 1 square. So you draw 4 squares, 1, 2, 3, and 4. We have the gene of one individual at the top. We're just looking at one gene now. We have lowercase m over 1 square, lowercase m over the other square. We put the other individual on the other side, and this individual's genotype is capitalized M and capitalized M, so homozygous dominant M. Now all we have to do to figure out what we're going to see in the offspring is to just bring these letters down into this square. And we always write the dominant gene by convention first. So we would write capital M, lowercase m, because I'm bringing this m and putting it here, and I'm bringing this m and putting it here. And then that would fill out this square. And to fill out this square over here, again, because I always write the dominant gene first, it would be capital M in this square, and lowercase m gets dropped here. And we already know that the phenotype, so now we have the genotype of these two, right? And it is heterozygous for that gene. This individual is also heterozygous for that gene. That means it has two different copies of the gene. But we know because the pigmented gene is dominant, that because this dominant gene is present, then pigmentation is going to be expressed. And so we know exactly what the phenotype of these individuals would be. So we can look at the genotype, which is heterozygous for the gene M, and we can see that this is going to result in a giraffe that is pigmented, here heterozygous, again, for the gene M, and we can see an individual which is pigmented. And we just fill in the 4 squares. Again, capital M here, and we bring down the uppercase M, and we get this individual's phenotype. Same thing over here. I bring in the capital M here, I bring in the lowercase m here, and I get this individual's phenotype. Okay, so this is a 1-gene Punnett square. And so all the offspring, when we look at them, are going to be heterozygous for the gene M. And all the offspring are going to be pigmented because they all have the dominant gene expressing. Okay? So this is really super important to know because, you know, here in this example we're using giraffes, right? And we're just looking at pigmentation. But what if this were a gene for, let's just pick something just randomly that would be important to know whether or not this is a gene for some version of cancer, right? And if you had the dominant gene, then you would actually express that cancer. Well, we would know that if we had an individual that was homozygous for the dominant gene and bred with an individual that was recessive for the dominant gene, then all of their offspring would express that cancer, right? So what's important to know when you're looking at a gene and you're trying to determine what the offspring are going to have is you need to know how that gene is going to express. Is it going to have a dominant and recessive type of expression? Okay? And we'll see that there are other types of expression that are possible as well. But we still use the Punnett square to determine them. All right. So now we're going to take those second generation that we had, those homozygous individuals, and we're going to crossbreed the two homozygous individuals. So this is called a cross 2. So here they're heterozygous. I'm sorry. Here they're heterozygous. So we have capital M and lowercase m for this one individual, and we're going to breed it to, again, another heterozygous, capital M, lowercase m. And when we fill in our 4-box Punnett square for one gene, we just bring the letters over. So capital M here, capital M here. We can see that this individual is homozygous dominant for the capital M gene, right? For the pigment gene. And here, this individual, we bring this capital M here and this lowercase m here. We can see this individual is going to be heterozygous, but it's still going to phenotypically express the dominant gene because the dominant gene is present. Let's see what this individual's genotype is. We bring over, remember we always write by convention the dominant gene first, so we bring down this capital M first, bring over this lowercase m, and this individual is also heterozygous, but phenotypically will express the pigment because of the dominant pigmented gene. And then this last individual, when we look at the genotype, again, we bring over the lowercase m here, and we bring down the lowercase m here, and we can see this individual is now homozygous recessive for the pigmented gene, and we know that that results in an individual that is phenotypically expressing albinoism, okay? And so when we cross two heterozygous individuals, we always end up with this same ratio, okay, with a dominant and recessive expression. We always end up with the genotype, remember these are the letters, the genotype being 1/4 will be homozygous dominant, as we can see here. Because, and we can say 1/4 because there's different ways to express the same thing. And this tends to confuse people because you can say it in different ways, but you're saying the same thing. So we have 4 boxes, so each box is a quarter, right? But each box is also a percentage, right? So if I were describing the genotype of the offspring here, I could say that 1/4 is homozygous dominant, right? Or I could say it as a percentage. 25% are homozygous dominant, right? Here we can see that 2/4 are going to be heterozygous. So 1, 2. So 2 of the 4 boxes, which is the same as 2/4, are heterozygous. And that is speaking of the genotype, right? I could also say the exact same thing in a different way. I could say 50% are heterozygous. So remember, when we're speaking of genotypes, we can either say it as a percentage, or we can also give the decimal equivalent of it, right? And here we have the same thing when we're looking at the genotype. 1/4, 1/4, are homozygous recessive, right? When we look at the phenotype, though, completely different, right? How that gene expresses in the offspring. So here we can see that phenotypically 3/4 of the offspring are going to be pigmented, right? Or I can say that in a different way. I could say 75% of the offspring are pigmented. And here we can see 1/4, 1/4, of the offspring are going to be albino, right? And I can say that in a different way, but say the exact same thing. I could say 25% will be phenotypically albino, okay? So remember that. So don't let that confuse you, because you will see it stated in different ways. You may see it stated as percentages, you may see it stated as fractions, you may see it stated as decimals, and it doesn't matter. It's still talking about these 4 boxes, okay? These quarters, all right? So don't let that confuse you. And also in different exams and questions, you have to pay attention to what they're asking you for. They may ask you what percentage are going to be albino. Well, now you have to give the percentage, right? And albino being, it's asking you the phenotype. So in that case, you would say, because it's asking for a percentage, you would say 25%, right? Here, again, a question may be, what fraction of the offspring will be homozygous dominant? So now it's asking you for a fraction. So you're stating it in the same way. So 1/4 of the offspring will be homozygous dominant, because now it's asking you for the genotype. Not the phenotype, what it looks like, but the actual genotype. So you have to pay attention to those 2 things in a question. Is it asking you for the genotype or the phenotype? And what method is it asking you to express it in? Decimal, fraction, percentage, right? They're all talking about the same thing. All right, here we go. So it turns out that genetics is very much probability. And so for the math majors out there, you're going to love this. So you could actually figure out the same thing using math. Most people do not like math. which is why the Punnett square makes it so much easier. However, when you get into looking at more than one gene, say you're looking at 3 genes or even 4 genes, the Punnett square, the use of the Punnett square can become very unwieldy. It can become very problematic because you're not just dealing with 4 squares, the number of squares that you're dealing with actually begins to expand. And so being able to use mathematics in order to find out your percentages of the offspring, looking at multiple genes, math becomes very, very helpful. And so here, what you can do if you want to determine what your genotype and your phenotype of the offspring would be mathematically, you would multiply the 2 components together to determine the overall probability, right? So here it's giving you an example of if the mother is albino and the father is heterozygous. So if the mother's albino, we know that she's homozygous recessive, right? Automatically, we just know that because it's a recessive gene, so she has to be homozygous in order to even express it. And the father is heterozygous, which means that he has both versions of the gene, right? And now there's 100% chance that the mother's egg will carry the recessive allele because that's all she can give. That's all she can put in an egg is the recessive allele. So 100% chance that her egg will carry the recessive allele and a 50% chance that a sperm will carry the recessive allele in the male because he could have one that has the dominant allele because he has the dominant allele. And he can have a sperm that has the recessive allele, and that's a 50% chance. And so now we can go ahead and do our multiplication, multiply the 2 components together to determine the overall probability. And so we end up with a 50% chance the offspring will be albino. So this is just another way to figure out your Punnett square mathematically, if you are more inclined to use mathematics or you're into more than one gene. Okay, so let's take a look at something like Tay-Sachs, which is a disease that can happen in humans. And here now we have each parent being heterozygous, meaning it has one of each copy of the gene. Each parent is heterozygous for Tay-Sachs. And now we know that there's a 50% chance that a gamete that he or she will carry will have the recessive allele, right? Because they could give one of each. One egg could be get the dominant gene. One egg could get the recessive gene version of the allele of the gene, version of the gene. Same thing with the male. One sperm could get the dominant version of the gene and the other sperm could get the recessive version. Now we want to try to figure out, well, what's the chance that the child of these heterozygous parents will be homozygous for Tay-Sachs and actually have and express the disease, which is Tay-Sachs disease, because that is how this is most commonly used when you have 2 parents who want to know what's the probability of my child actually developing this disease that both parents are heterozygous for. Because Tay-Sachs only expresses if you're homozygous recessive, then both the parents are going to appear normal. They are not going to express Tay-Sachs. They're not going to have Tay-Sachs disease, yet they carry the gene, okay? And so here we can do our multiplication again, right? So there's a 50% chance that a gamete that he or she produces will carry the recessive allele. Now we multiply the 2 components together, so 0.5 times 0.5 to determine the overall probability, and we end up with a 0.25 or 25% chance that the child of these 2 heterozygous parents will be homozygous for Tay-Sachs, okay? And again, you can work that out in the Punnett square, but this is a mathematical way of looking at the same thing, okay? So what are test crosses? So let's take a look at some white alligators and figure out what a test cross is. So we can use a test cross when we're trying to determine the genotype of an unknown individual. We have an individual, we don't know what their genotype is. No idea. can't tell. What we do is what's called a test cross. So here we have the homozygous recessive individual, and we cross that homozygous recessive individual with this unknown individual. So the unknown individual has pigment. And what we don't know is this individual homozygous dominant because they have pigment, or is this individual heterozygous because they also have pigment, right? And so now we're trying to figure out, are they homozygous dominant or are they heterozygous? We don't know. What you do is a test cross with a homozygous recessive. And what happens? So if we do a test cross with a homozygous recessive, if the individual is homozygous dominant, this unknown individual, if the individual is homozygous dominant, then all the offspring will be pigmented, right? Because they're all going to get the dominant gene. That's all the individual could give if it's homozygous dominant. And so all of the offspring are going to express with pigment. However, if it turns out that our unknown individual in our test cross ends up having offspring that are albino or basically homozygous recessive, then we know this unknown individual was heterozygous, right? Because somehow, somewhere in his genotype, there was the recessive gene that we could not see phenotypically because the dominant gene is going to express. However, the recessive allele will end up somewhere in the offspring. And when we see that, then we know that this individual was heterozygous. Okay, so that's what a test cross is, so that you can try to determine what an unknown individual's genotype is for a gene that you are interested in. All right. I wanted to give a little mind break because this can get very deep and it's going to take a while for you to work through some Punnett squares to actually get comfortable with one gene Punnett squares. I do want you to go ahead and do that. I've got some sites that you can go to practice at. Also, one of your interactives for this week is working with Punnett squares. The more you work with them, the more comfortable you get with them. But it gives us all the diversity. We can understand all the diversity that we start to see on this planet. So we'll take a mind break and look at a little diversity on the planet. This is actually a contest that Nikon has every year called Nikon Small World, and it's a photography competition looking at very, very small things. And some amazing photographs are taken by some photographers that are out there. This is actually looking at the eye of a beetle, which is amazing, right? And here's another beetle that was submitted as part of that contest. Really, really beautiful. A lot of these insects really just remind me of a lot of movies that we see where the creators of the movies kind of use them as inspiration. This reminds me sort of like the movie Men in Black, where they had different aliens that looked very much like this little guy that we have. And I just wanted to show you one other. This is a wasp. Just fantastic photography, taking a look at them and just showing how amazing they are and how beautiful they are when you look at them up close. But look at the mouth, look at the mandible. That's pretty scary. But there's beauty in nature, right? All right, back to work. So we're going to look at a pedigree and understanding how to read a pedigree. So a pedigree is a really useful tool to document a trait of interest across multiple generations of family members. And this is used a lot, especially in the cancer setting, where genetic counselors are trying to determine, basically sort of tracing cancer through a family line. So here's how we actually read a pedigree. Um, a circle means that the individual is female. Okay? And if a circle is filled in, that means that the individual is exhibiting that trait of interest, whatever trait it is you are tracing or you're looking at. Okay? So if it's filled in, then that individual is exhibiting the trait. If it's not filled in, that means that individual is not exhibiting the trait of interest. A square signifies a male. And again, if the square is filled in, then that means the male exhibits the trait of interest. And if the square is not filled in, that means the male does not exhibit the trait of interest. So now you go through and you ask the individual about their parents, right? So here we have the grandfather and here we have the grandmother. We can see the grandfather had the trait of interest. Let's just say cancer, okay? Here we can see the grandfather had cancer. The grandmother did not. When we start to look at their offspring and we come down, we can see that the aunt had cancer. The uncle did not, right? And the father of the person who we are speaking to had cancer, right? And the mother of the person that we are speaking to did not, right? And so we start to fill in these squares just by asking this person questions about the family tree. And so it helps us, it's a tool, it helps us understand the genotype that is exhibited throughout the family. So here we can see, let's start with the individual here. The individual has two sisters. One of the sisters has cancer. When we go back to the father, we can see that the father had cancer, the mother did not. When we go to the mother's parents, we can see they did not have cancer, neither did any of their siblings. When we go to the father's side, because the sister had cancer, right? We go to the father's side of the family, we can see that the father's brother did not have the cancer, but the father's sister did have cancer. And when we go back to the father's parents, we can see the grandfather had cancer, right? And we can see that the father's sister also had children, and the first cousin of this individual developed cancer as well. Okay, so we can begin to trace a gene throughout a family line using a pedigree. All right, that's how you read a pedigree chart. So this is a trait that you'll see very, very commonly in dogs. This trait is actually bred for. In hunting dogs, it's called anery. It's a condition in which dogs and other animals, they're born without a tail. And the condition is inherited as a recessive trait. Okay, so let's look at a pedigree and see if we can understand that. So it's a recessive trait. All right, so here we have this pedigree. So if a dog has no tail, but both parents do, both parents have tails. So what we don't know about the parents yet is, okay, they have a tail. So does that mean that they're a homozygous dominant or they're heterozygous? Okay, but the dog has no tail, right? So we know that he had to get that recessive gene from both parents, right? So now we can begin to fill in this pedigree. So this puppy definitely inherits a recessive allele from its father. It will have anery. If, one, its mother has a recessive allele and she passes that allele on to the puppy, what is the probability? Okay, so here again, we're going to read our pedigree chart. Remember, females are circles, and if it's filled in, that means that they do not have a tail, they have anery. If it's empty, it means that they have a tail. They're not exhibiting anery. Okay, filled, no tail. Empty, tail. Male is a square. Filled, no tail. Empty, has a tail. And so now we can begin to read the chart. So here we have the parents here. right? And we can see that here is the female, not filled, she has a tail. Here is the male, not filled, he has a tail. However, we have two offspring here. One is a male, and it does not have a tail. We come over here, we have a female, and she has a tail. Now, here we have another box, and we have a male, and he does not have a tail. And the question is, now I am breeding this male who does not have a tail. We know that the gene is homozygous recessive. In order to not have a tail, you have to be homozygous recessive, right? So we know this individual here is homozygous recessive. And this individual is breeding with this individual here. And the question is, well, what is the genotype of this individual? It came from two heterozygous parents. Okay? And we know that because when these two individuals who both have tails bred, they ended up with a puppy that didn't have a tail, which means they both were heterozygous. There was that recessive gene hiding in there. Had to be for this puppy to show up. Now the question is, what is the genotype of this individual, right? So you have to do the Punnett square in order to figure that out, right? But that's how we can use our pedigrees to actually take a look and follow family trees for different genes, and we can use our Punnett square to determine the probability of different individuals, and it is extremely, extremely helpful. So please work on figuring out how to use your Punnett square. Practice that. It's going to be very important. Okay. I want to take another mind break because I know that was a lot, and we will kind of jump into some current events in science. So this actually was in 2019. Remember that? 2019 was a long time ago, right? In 2019, governments were banning face masks in order to enable facial recognition software. Of course, now they can't do that because there is a pandemic. But I wanted to take just a moment to break away to explain to you what facial recognition software is, that it's not this magic that a lot of people think that it is. And here, what you are seeing in this image is basically the face of an individual. Certain landmarks are actually laid out in the data, and we can see that basically what's happening is the computer through the algorithms are just matching the landmarks of the face to a landmark, the same landmarks of the face that is in its database. And so this really is nothing more than matrix multiplication in order to do that. Okay. And so I just wanted to show you that because it is nothing more than math, it is algorithms, it is matrix multiplication, that the recognition software is actually using, it can very easily be fooled. And here, this is one lab that works on machine learning. And this is an adversarial lab, defeating machine learning algorithms. It's a specialty, which is very interesting. And it seems like it's a pretty fun specialty. And what they've come up with is there is this image here, which looks to us very much like just a party scene, but it very much fools the algorithm. And so when that image is used, the algorithm doesn't know it's a person. You can see he just turned around and the algorithm's like, oh, well, there's a person now. And when he turns it back, the algorithm's like, yeah, don't see him. All I see is that party image, and I don't recognize that as a person. So it's pretty cool. And so there are different labs that work on what's called adversarial training, understanding how algorithms can be fooled. Here's one where, again, they came up with these adversarial glasses, which look kind of strange, right? They just look like yellow glasses with some stripes on them. But it turns out when you're wearing these glasses with these different stripes, because of the training data, the facial recognition software will think you are certain celebrities, which is so funny. You won't come up registered as who you are. It will come up that you are this individual, or if you're wearing this version of the glasses, it will tag you as this individual, or if you're wearing this version of the glasses, it will recognize you as this individual. So it's actually pretty funny and easily to fool. Okay. So let's jump back into the science. Okay. So now I explained to you that there was such a thing as dominant and recessive genes, right? Where there's other ways of gene expression. So there is what's called incomplete dominance. And here's a way I like to remember incomplete dominance. See this INC in incomplete? When you see incomplete dominance, it means that the heterozygous version will be a mixture of the two, like ink, like you mixed ink, right? So here we have, let's just say, we have the father, we're looking at plants here, the father is a red-flowered homozygous. And again, when we have these different expressions of the gene, such as incomplete, then we write them in superscript. Okay, that way, again, by convention, when we look at a gene and we see that it's in superscript, we know that it's not just a straight dominant and recessive expression of the gene, that we have a different type of expression going on here. And so we're looking at the superscript of the gene, and here we have homozygous red mixing with, breeding with a homozygous white, and because it's incomplete, like ink, then the heterozygous offspring are going to be pink. Okay? So red is not completely expressing, white is not completely expressing. It is a combination of the two genes. So we mix the red and the white, and because it's incomplete dominance, we end up with pink offspring. And if we were to take those pink offspring and we were to breed them together, then we would start to see our genotypes come out, right? We can see the white flower, and we would get two of our heterozygotes again, and we would get the red again, right? Because of the gene expression. So same thing, because neither allele is dominant, we cannot use uppercase and lowercase for the allele. Instead, the superscript W, which is being used here for this gene, represents the allele that produces the white flower, and R represents the red flower allele. And so the discoverers of the individual genes that we're talking about will give them the letter that that will signify that gene. So I don't want you to think there's some magic with W always represents this or R always represents that. It isn't. So you actually would have to know, you'd have to look up what type of expression that gene has and what letter is being used for the different alleles of that gene, okay? But if it's not just a straight dominant and recessive expression, you're going to see it written in superscript, and that tells you that it may be incomplete dominance, okay? Which is like ink. When we get heterozygous, it's a combination of the two. We get pink. But there's another type of expression that's possible. This is called codominant expression. And this means that instead of getting a mixture of the two, red and white, I get pink. Codominance means that they're both expressed, which means if I have red and white, I'm going to get a red and white stripe, right? Which is what's sort of being illustrated here. You know, one gene is going this way and the other gene is going this way. Well, if it's a codominant expression, then we're going to see the offspring express both of them equally, right? Which is different than incomplete. Incomplete is a mixture, right? Okay, so we have incomplete expression and codominant expression. All right. So now we're going to talk very quickly about multiple allelism. This is what we see with blood and blood types. Blood types are a little bit different. So multiple allelism occurs when there are 3 or more alleles for a gene within a population. So an individual still inherits only 2 alleles, 1 from each parent, right? You can still only inherit 2, 1 from each parent. All right. But here, 3 alleles are possible for this gene. We could have an A allele, we could have a B allele, or we could have what's known as an I allele. Okay? And A is dominant to I, but it's codominant with B. And B is dominant to I, and it's codominant with A. And I is recessive to both A and B. Okay? So, what are the 6 possible genotypes that we can have? So we can have someone with type A blood. If you have type A blood, you can either have AA or you can have AI. They're both going to express as type A. Why? Because I is recessive to both A and B. So if A is there, it's going to be expressed. So someone with type A blood could be either AA or AI. Someone with type B blood can be either BB or BI, right? Someone with type AB blood can only be AB, right? Because you only have one of 2 genes. You receive 1 gene from each parent, right? Someone with type O is II, okay? And that's how blood types actually work. And let's take just a little bit closer look at why we have A, B, and O, what that actually means. All right. So, antigens, as you know, are proteins on the surface of some cells. They act as signposts that tell the immune system whether the cell belongs in the body. So if you have type A blood, you're expressing A antigens, and individuals, as we know, we produce antibodies, and individuals produce antibodies to the antigens that they don't have on their cells. So if you have type A blood, you have type A antigens, and you will have antibodies against type B, right? If you have type B blood, you're going to express type B antigens, and you will have antibodies against type A, right? If you have type AB blood, then you will express both A and B antigens, and you will not have any antigens or any antibodies against A or B. So what does that mean? So that means if you're type A and you have antibodies against type B, somebody gives you type B blood, you're going to destroy it. You're going to have a severe immune reaction. The body's going to think you're being attacked by type B blood, right? That's not good. If you have type B blood, because you have B antigens on your blood, you're going to have antibodies against type A blood. If somebody gives you type A blood, you're going to have this huge reaction, right? Because you have antibodies against A because you are type B. If you are type B, you don't have any antibodies against either one, so you are the universal acceptor. You can accept blood from A and B and O, right? So anybody can donate blood to you, which is really cool. Now, if you are type O, you don't have any antigens on your red blood cell surface. You don't have A antigens, you don't have B antigens. However, you have antibodies against A and B. So if you're given type A blood, you're going to have a huge reaction. If you're given type B blood, you're going to have a huge reaction, right? You can only accept type O blood. But you are the universal donor because you don't have any antigens, and so you can donate to type A, you can donate to type B, you can donate to type A and B. So O is the universal donor, okay? So basically that's what this is showing you here. So type AB blood can accept from anyone. Type AB blood can accept from type A, can accept from type B, can accept from type AB, can accept from type O, right? Type O can donate to anyone. However, type O can only receive from type O. Okay. All right. So that is how blood typing works. Make sure you go through that chart and you understand it, because that's important to know. It's really, really important to know. It affects your life moving forward, so you want to make sure you understand it. All right, let's move on with genotype to phenotype. So there is something called polygenic traits, and that is, that means that poly meaning many. We can have something called pleiotropy. One gene, lots of effects. So it's not like you have one gene and it only has this one effect. That's not always the case. Okay, remember, in biology and science, things can get a little bit more complicated, right? So pleiotropy can occur when one gene can influence multiple different traits. For example, let's take a look at sickle cell anemia. Here we have an individual who is homozygous, and they do not have the sickle cell disease trait at all. This individual is susceptible to malaria. Malaria will kill this individual. Here is the malarial organism here infecting a red blood cell. Someone with no sickle cell does not have the ability to sickle their cells, and so malaria can enter the red blood cells and basically burst them open, and that's how you die of malaria. Now, through adaptation and evolution, sickle cell is actually an adaptation to survive malaria. And so we can have an individual who is heterozygous for the gene. We can see here with an S in superscript. That will cause the cells to sickle. This individual does not have what is considered sickle cell disease, but this individual is immune to malaria. A portion of their red blood cells will sickle. Okay. And then we have someone who is homozygous. We can see superscript Fs in both. This individual has sickle cell disease. This individual is immune to malaria, but generally during their first sickle cell crisis, because all of their red blood cells will sickle, that's not conducive to life because you're not carrying enough oxygen. Usually these individuals will die during their first sickle cell crisis. So it is these heterozygous individuals that go on to keep a population that is immune to malaria. And there are different parts of the world where malaria, which is carried by a mosquito, Africa being one of them, it's very prevalent, right? And in order to survive without drugs and things like that and medications, which are not available, then nature has actually evolved a way to survive malaria. And it is through this sickle cell trait, okay? And this is an example of pleiotropy, one gene, but many effects, okay? Let's take a look at sex-linked traits. So we're looking at different ways that traits can actually have, or genes can have expression. So a sex-linked trait is carried, for example, on, let's say, the X chromosome. And so women can carry 2 copies of the X chromosome, while men carry an X chromosome and a Y. So they have 1 copy. So here we go. So let's say we have a gene with instructions for light-sensitive proteins within the eye. And let's just call this gene R. So we have uppercase R and lowercase r. And so 2 alleles are possible, right? We're looking here at our X gene. So if you have the uppercase R, then this produces functioning light-sensitive proteins. Okay, if you have lowercase r, this is going to produce defective light-sensitive proteins. Now, in order to be colorblind, this is how color blindness actually occurs. In order to be colorblind, a male must inherit a color blindness allele, which is lowercase r, from his mother, because that's carried on the X chromosome, right? That's who he gets his X chromosome from, right? And a female must inherit color blindness from both parents because she has two X's, right? And basically, she would have to get both alleles from both parents in order to be colorblind. But in order to have normal vision, okay, then a male must inherit a normal color vision allele from his mother, here, as we can see. And a female can inherit a normal color vision allele from either her mother or her father. So even though this recessive allele is present, because there's the dominant allele, this person's not going to be colorblind. And here, we have homozygous of the allele as well. So this person is not going to be colorblind. But because it is not on, there's no gene that is producing this functioning light-sensitive protein. It's not carried on the Y chromosome. It's only carried on the X. That we can see how mostly males are affected with this condition, right? This is why mostly males tend to be colorblind, right? Because if you're female, you have two X's, and if you get a light-producing copy, which is the dominant one, it's going to override the other one, and you're going to be able to actually see color, right? But if you're male, you only have one X chromosome, so either that's one that's going to produce that light-sensitive protein or not. And if you have the one that does not, then you will be colorblind, okay? And that's why we see it mostly in males. Okay, that is sex-linked traits. Explaining that. Okay, so I wanted to explain this. If you look at a can of soda, here this is Diet Coke, it has this warning that you probably have never seen, but if you have this disease, you certainly look for this warning. And it says phenylketonuria. This contains phenylalanine. And that is there, and it is on every can of soda that may contain phenylalanine, because the ingestion of phenylalanine can be fatal to individuals with the inherited condition phenylketonuria, PKU. And there is a warning label on products that actually have phenylalanine in them in order to try to protect them. So this is to protect individuals that have this genetic predisposition. Okay. But here we have some pigment genes produce dark pigment only under cold conditions. So we're looking at how genes can actually express, right, their expression. And another way is some pigment genes can produce dark pigment only under cold conditions, such as on the tail, the nose, the ears, and the feet. And we can see this expressed in cats that give us this lovely color here, coloration of the cats. And also we see it in rabbits. Okay, and the expression is based on the temperature of the region, which is very, very interesting. All right, so I want to kind of quickly go over Mendel's law of independent assortment. So Mendel's law of independent assortment states that one trait does not influence the inheritance of another trait. So if parents are both heterozygous for both traits, okay, then 4 different types of gametes are produced by each, right? So this is now looking at 2 different traits. Before we looked at 1 gene Punnett squares. Now you're looking at a 2 gene Punnett square, okay? So we're looking at here, the mother is short hair, heterozygous for that gene for short hair, And then she is also heterozygous for the gene for white fur. So we're looking at 2 genes here, right? And then we start to look at the possible gametes that can be produced. Because remember, that's what we put across this top line and this side line, the possible gametes that this one individual can produce and the possible gametes that this other individual can produce. And then we start filling in the squares. And when you are working with 2 genes, you have to make 16 boxes. 1, 2, 3, 4. 1, 2, 3, 4. 4 times 4 is 16. So there's 16 boxes here, right? And so when we look at the possible gametes that can be produced by 2 genes, then when the individual is heterozygous for one and heterozygous for the other, same thing here. The father is heterozygous for the gene for short hair and heterozygous for the gene for white fur. Then we have all of his possible gametes that can be made here. And then we start basically filling in our boxes for the genotypes, and then we figure out from the genotypes what is our phenotypic expression of the individuals. And Mendel's law says that Mendel's law of independent assortment states that one trait does not influence the inheritance of another trait. Okay, so your boxes are still going to be filled in the same. Okay, that's basically what that is saying. Okay, let's take a look at linked genes. What does that mean? So another type of expression. So here we have a paternal copy and a maternal copy of genes. So we're looking at homologous chromosomes. And we can have what's called a linked gene. So when crossing over occurs, remember when we have that recombination that's happening, linked genes usually stay together. So they usually will cross over together. So it's 2 genes that will move together, okay? That is called linked genes. So if genes are really far apart on a chromosome, they can be on different chromosomes, the inheritance of one gene does not influence the inheritance of another. But we can have linked genes, which means that during recombination, they will usually stay together. So what does that mean? That means that typically we'll see different phenotypic expressions happening together. Red hair and freckles. Those 2 genes are linked, they move together. Okay, that's really all that means. Super simple. We've seen it before. All right, that's going to conclude this lecture. I'm going to have this link to do a study, a little Kahoot that's kind of fun for you to work through and kind of test yourself. I will have this link inside of your module for this week. So make sure after you do your studying and you've practiced your Punnett squares, you've done your interactive, because that's going to help you with the Punnett squares as well, especially your 2 gene Punnett squares. Remember, we are looking at the gametes. That's the most important part when you're working with 2 gene Punnett squares, figuring out what can be in each gamete. I will try to find a really good video to kind of help you guys out with that, making 2 gene Punnett squares. I'll see if I can find a good one to put in to help you with that. All right, but make sure you play this little Kahoot. Get yourself all prepared for your exam 2 that's going to be coming up. And I will see you guys next time. Bye.